Something happens to your eyes while you sleep that most people have never been told about. Not the obvious part, not the rapid eye movement, not the dreams, not the fact that your eyes are closed and resting. Something more specific than that. Something that happens at the cellular level, in the tissue at the back of your eye, in the hours between midnight and early morning, that determines, to a degree that the ophthalmology literature has been documenting with increasing clarity for 2 decades, whether your vision at 70 looks like your vision at 60, or whether it begins the kind of gradual, irreversible decline that most people have been told is simply inevitable. And the reason most people have never heard about it is partly because ophthalmology appointments are short, and explaining cellular biology takes time. Partly because the intervention I am going to describe today does not generate pharmaceutical revenue and therefore does not receive the kind of sustained public communication that medications do. And partly because the idea that what you eat before bed has a direct and measurable influence on what happens inside your eyes while you sleep sounds, on first hearing, almost too specific to be credible. It is credible. The mechanism is real, the evidence is consistent. And by the end of today, you are going to understand exactly what is happening inside your eyes at night, why it is relevant to every major cause of vision loss in adults over 60, which food acts directly on that process, and how to use it correctly. Because the how matters as much as the what, and most people who have encountered pieces of this information have not had the full picture. Let us start with the back of your eye. The retina is the light-sensitive tissue that lines the inner surface of the back of the eyeball. It is the structure that converts the light entering your eye into the electrical signals that your brain interprets as vision. It contains 2 types of photoreceptor cells: rods, which are responsible for peripheral and low-light vision, and cones, which are responsible for central vision and color discrimination. The concentration of cones is highest in a small central region of the retina called the macula, roughly 5 millimeters in diameter. And within the macula, the highest density of all is at the very center, a tiny pit called the fovea, which is responsible for the sharp central vision you use to read, to recognize faces, and to see fine detail. The macula is the most metabolically active tissue in the human body, relative to its size. The photoreceptors within it have an extraordinarily high energy demand. They are constantly cycling through a process of light absorption, signal generation, and molecular regeneration that consumes more oxygen and produces more oxidative byproduct per unit volume than virtually any other tissue. That metabolic intensity makes the macula simultaneously the most visually critical and the most biologically vulnerable region of the eye. Beneath the photoreceptors lies a single layer of cells called the retinal pigment epithelium, the RPE. The RPE performs one of the most remarkable and least discussed functions in the human body. Every day, the outer segments of the photoreceptors, the parts of the rods and cones that actually absorb light, are shed and must be replaced. This is a continuous renewal process that the photoreceptors cannot perform alone. The RPE engulfs the shed outer segments, digests and recycles their components, and simultaneously maintains the health of the photoreceptors above and the choroidal blood supply below. It manages the transport of nutrients from the blood into the photoreceptor layer and the removal of waste products in the opposite direction. It produces growth factors that support photoreceptor survival, and it is the site of the visual cycle, the biochemical process by which the light-sensitive molecules in photoreceptors are regenerated after each photon absorption event. The RPE is, in other words, the caretaker of the most critical tissue in your visual system. When it functions well, the photoreceptors above it survive, renew, and function. When it fails, when oxidative damage, lipid accumulation, or inflammatory injury compromise its function, the photoreceptors above it begin to die, and they do not come back. This is the biology of age-related macular degeneration, the most common cause of irreversible central vision loss in adults over 60 in the developed world. And it begins not with the dramatic vision loss that characterizes advanced disease, but with the slow, accumulating failure of RPE function over years. Driven by oxidative stress, by chronic low-grade inflammation, by the accumulation of lipid waste products called drusen beneath the RPE, and by the compromised blood flow through the choroidal circulation that supplies the RPE with the oxygen and nutrients it needs to sustain its extraordinary metabolic workload. Now here is where sleep becomes directly relevant to this biology. And this is the part that is almost never discussed clearly. The choroidal circulation, the network of blood vessels that lies beneath the RPE and supplies the outer retina, operates differently at night than during the day. During waking hours, with constant light stimulation of the photoreceptors, creating sustained high metabolic demand, the choroidal blood flow is directed primarily toward meeting that demand. The tissue is in a state of continuous active function, consuming oxygen and nutrients, generating metabolic waste, and relying on the RPE to manage the interface between blood supply and photoreceptor need. During sleep, particularly during the deeper stages of non-REM sleep, several things happen simultaneously that are uniquely important for retinal health. The light stimulus to the photoreceptors ceases, the immediate metabolic demand decreases. The circadian-controlled vasodilation of the choroidal vessels increases blood flow to the outer retina during specific windows of the sleep period, particularly in the early morning hours. And critically, the glymphatic system, the brain's waste clearance mechanism that operates primarily during sleep, extends its function to include the structures of the visual pathway, including the retina and the optic nerve. The glymphatic system, discovered in its modern form by Maiken Nedergaard and colleagues at the University of Rochester in 2013 and published in Science, is a network of fluid channels surrounding the brain's blood vessels through which cerebrospinal fluid circulates during sleep, washing out the metabolic waste products that accumulate during waking hours. The clearance of amyloid beta, the protein whose accumulation is central to Alzheimer's disease pathology, occurs predominantly through this glymphatic pathway during sleep. And subsequent research, including a landmark 2019 study published in Current Biology, demonstrated that glymphatic clearance extends to the retina. That the retina has its own glymphatic-like system that clears waste products, including the toxic byproducts of the visual cycle and oxidized lipids that contribute to drusen formation, predominantly during sleep. This is the biology that most people watching this have never been told. Your eyes are not simply resting during sleep. They are being cleaned. The toxic accumulations that drive RPE damage, drusen formation, and ultimately macular degeneration are being cleared, or failing to be cleared adequately, during the hours when you are asleep. The quality of that clearance, the adequacy of the choroidal blood flow supporting it, and the metabolic environment in which the RPE performs its renewal work during the sleep period are all directly influenced by the nutritional status of the retinal tissue. And the nutritional factor that most directly and specifically supports this nocturnal retinal biology, the one with the most consistent evidence for protecting RPE function, reducing oxidative stress in photoreceptor tissue, and supporting the clearance of the lipid waste products that precede macular degeneration, is a pair of carotenoids called lutein and zeaxanthin, specifically cooked spinach or cooked kale. I want to explain the mechanism with the same precision I have applied to everything else today, because this is not a case of a food having a vague general health benefit that someone has stretched to apply to vision. The mechanism is specific, well-characterized, and directly connected to the biology I have described. Lutein and zeaxanthin are the only dietary carotenoids that selectively accumulate in the retina. Of all the carotenoids present in human blood, and there are many, including beta-carotene, lycopene, and alpha-carotene, only lutein and zeaxanthin cross the blood-retina barrier and concentrate specifically in the macular region. This selectivity is not accidental. It reflects a specific transport system involving a protein called STAR-D3, or steroidogenic acute regulatory domain protein 3, that actively pulls lutein and zeaxanthin out of the circulation and deposits them preferentially in the macula, where they form what is called the macular pigment. The macular pigment is not simply a passive structural feature. It is an active, multifunctional protective system for the most vulnerable region of the visual apparatus. The first function is optical filtration. Lutein and zeaxanthin absorb short-wavelength blue light, the wavelength range most energetically damaging to the photoreceptors and most capable of initiating the oxidative cascade that damages the RPE. In an era of chronic screen exposure and LED lighting, both of which are enriched in the blue wavelengths that the macular pigment filters, this function has become more relevant, not less. The second function is direct antioxidant activity. Lutein and zeaxanthin are highly effective quenchers of singlet oxygen, the specific reactive oxygen species generated in highest concentration in the retina during light exposure through a photosensitized oxidation of the lipid-rich outer segments of the photoreceptors. The retina, as the most metabolically active tissue per unit volume in the body, generates singlet oxygen at rates that would destroy its own cells without adequate antioxidant protection. Lutein and zeaxanthin, positioned within the membrane structures of the photoreceptors and RPE, quench this oxidative threat at the point of generation, not after it has had time to initiate the lipid peroxidation cascade that damages cellular membranes. The third function is anti-inflammatory signaling. Beyond their antioxidant role, lutein and zeaxanthin modulate the NF-κB inflammatory signaling pathway within retinal tissue. the same master inflammatory switch relevant to cardiovascular and cognitive disease, reducing the production of pro-inflammatory cytokines within the RPE in the subretinal space that contribute to the complement-mediated inflammation central to macular degeneration pathogenesis. The fourth function, and the one most directly relevant to the nocturnal retinal biology I described, is support of the visual cycle and RPE function during the sleep period. The regeneration of the visual pigment rhodopsin in rods and the cone photopigments, the biochemical process that must occur during darkness to prepare the photoreceptors for the next day of light exposure, produces oxidative intermediates that stress the RPE cells managing the process. Adequate lutein and zeaxanthin within the RPE membrane reduces the oxidative burden of this nocturnal renewal process and supports the integrity of the RPE cells performing it. The clearance of the oxidized lipid waste products, the lipofuscin and drusen precursors that accumulate in the RPE and beneath it with age, is influenced by the oxidative environment in which that clearance occurs. Better antioxidant protection during the nocturnal clearance window means more efficient removal of the waste products whose accumulation is the earliest measurable precursor to macular degeneration. This is the mechanistic chain connecting dietary lutein and zeaxanthin consumption to what happens inside your eyes while you sleep. It is not a loose association. It is a sequence of specific documented biological steps. Now let me tell you what the clinical evidence shows. The Age-Related Eye Disease Study 2, ARDS-2, is the largest and most rigorously conducted randomized controlled trial in the history of nutritional ophthalmology. It enrolled over 4,000 participants with intermediate or advanced age-related macular degeneration in one eye and followed them for 5 years, comparing the effect of supplementation with lutein and zeaxanthin versus beta-carotene, alongside vitamin C and E and zinc, on the progression of macular degeneration to advanced disease. The results, published in JAMA in 2013, found that participants receiving lutein and zeaxanthin had a statistically significant reduction in the risk of progression to advanced macular degeneration compared to those receiving beta-carotene, with the benefit most pronounced in participants whose baseline dietary intake of lutein and zeaxanthin was lowest. That final finding deserves emphasis. The benefit was greatest in those who were most deficient, and the most deficient participants were those eating the fewest dark leafy greens, the primary dietary source of lutein and zeaxanthin in the Western diet. The trial confirmed that lutein and zeaxanthin are the carotenoids most relevant to macular protection, displaced beta-carotene from the formulation on the basis of the evidence, and established a direct relationship between these specific nutrients and the rate of disease progression in the most common cause of irreversible vision loss in older adults. A 2017 study published in Investigative Ophthalmology and Visual Science examined the relationship between dietary lutein and zeaxanthin intake and macular pigment optical density, the measurable concentration of lutein and zeaxanthin in the macula, in adults over 60. Higher dietary intake from food sources, particularly dark leafy greens, was associated with significantly higher macular pigment optical density, and higher macular pigment optical density was associated with better contrast sensitivity, better performance in low light conditions, and reduced photostress recovery time, the time the eye requires to recover visual function after brief exposure to a bright light stimulus. Each of these functional measures declines with age in most people, and the magnitude of that decline was significantly smaller in individuals with higher macular pigment optical density. A 2019 systematic review published in Nutrients analyzed 12 randomized controlled trials of lutein and zeaxanthin supplementation across multiple visual endpoints in older adults and found consistent improvements in macular pigment optical density, contrast sensitivity, and glare sensitivity with supplementation, with the effects becoming measurable within 4 to 6 months of consistent intake. Beyond macular degeneration, the evidence extends to other major causes of vision loss in older adults. Cataracts, the clouding of the crystalline lens of the eye that is the leading cause of reversible vision impairment in this age group globally, developed through a process of oxidative modification of the lens proteins, which are extraordinarily long-lived and accumulate oxidative damage over decades of exposure. Lutein and zeaxanthin are present in the lens as well as the retina, deposited there by the same selective transport system, and provide antioxidant protection against the lens protein oxidation that initiates cataract formation. A 2008 study published in Archives of Ophthalmology, analyzing data from the Nurses' Health Study and the Health Professionals Follow-Up Study, combined over 100,000 participants followed for over a decade, found that higher dietary lutein and zeaxanthin intake was associated with a significant reduction in the risk of cataract extraction, with the association strongest in the highest intake quintile. Glaucoma, the progressive optic nerve damage that is the leading cause of irreversible blindness in adults over 60 worldwide, involves the death of retinal ganglion cells, the neurons whose axons form the optic nerve. Oxidative stress and neuroinflammation are recognized contributors to retinal ganglion cell death and glaucoma pathogenesis. Lutein has been shown in animal models and in emerging human observational data to reduce oxidative stress in retinal ganglion cells and to attenuate the inflammatory signaling that contributes to their degeneration. While the evidence in glaucoma is less mature than in macular degeneration, the directional consistency across the available data is meaningful. Diabetic retinopathy, the vascular complication of diabetes that is the leading cause of new blindness in working-age adults and that affects a significant proportion of older diabetic adults, involves both microvascular damage and neurodegeneration in the retina, with oxidative stress and inflammatory signaling playing central roles in both components. Lutein has been shown in multiple experimental models to reduce the expression of vascular endothelial growth factor, the molecule that drives the abnormal blood vessel growth of proliferative diabetic retinopathy, and to attenuate inflammatory cytokine production in retinal tissue exposed to high glucose conditions. The clinical evidence is emerging rather than definitive, but the mechanistic rationale is well grounded. The breadth of evidence across multiple distinct pathological processes, macular degeneration, cataracts, glaucoma, diabetic retinopathy, all pointing toward the same nutrients through converging but distinct mechanisms, is one of the strongest indicators that the protection being provided is genuine and broad, rather than artificially specific to one disease model. The breadth of evidence across multiple distinct pathological processes, macular degeneration, cataracts, glaucoma, diabetic retinopathy, all pointing toward the same nutrients through converging but distinct mechanisms, is one of the strongest indicators that the protection being provided is genuine and broad, rather than artificially specific to one disease model. Lutein and zeaxanthin are fat-soluble carotenoids. Like all fat-soluble compounds, their absorption from the digestive tract is dependent on the simultaneous presence of dietary fat, specifically, fat sufficient to stimulate bile acid secretion and the formation of the mixed micelles through which fat-soluble nutrients are absorbed across the intestinal wall. Consuming lutein and zeaxanthin from food in the complete absence of dietary fat produces dramatically lower absorption, some studies suggesting as little as a fraction of what is achieved in the presence of fat. Cooking dark leafy greens, rather than eating them raw, increases the bioavailability of lutein and zeaxanthin by breaking down the plant cell wall matrix that encases the carotenoids in uncooked leaves, making them more accessible to digestive processing. The combination of cooked dark leafy greens consumed with a source of healthy fat, olive oil, avocado, eggs, a small amount of cheese, delivers lutein and zeaxanthin in the form that the research most consistently associates with meaningful increases in circulating carotenoid levels and macular pigment optical density. This is not a minor detail. Studies comparing blood lutein levels after consumption of raw versus cooked greens, with and without fat, show differences in absorption efficiency that translate directly into differences in the magnitude of retinal deposition. The combination of cooked dark leafy greens consumed with a source of healthy fat, olive oil, avocado, eggs, a small amount of cheese, delivers lutein and zeaxanthin in the form that the research most consistently associates with meaningful increases in circulating carotenoid levels and macular pigment optical density. This is not a minor detail. Studies comparing blood lutein levels after consumption of raw versus cooked greens, with and without fat, show differences in absorption efficiency that translate directly into differences in the magnitude of retinal deposition. Spinach and kale are the two most concentrated common dietary sources of lutein and zeaxanthin in the Western food supply. A half cup of cooked spinach provides approximately 10 to 12 milligrams of lutein and zeaxanthin combined. A half cup of cooked kale provides approximately 10 milligrams. The levels associated with meaningful macular pigment increase in clinical trials range from 6 to 20 milligrams per day, meaning that a single serving of cooked spinach or kale with olive oil meets or approaches the threshold supported by the research. A single large egg yolk contains approximately 200 to 250 micrograms of lutein and zeaxanthin, far less by absolute measure than dark leafy greens. But the bioavailability of lutein from egg yolk is substantially higher than from vegetable sources because the lutein in the yolk is embedded in a fat matrix that facilitates absorption. Studies directly comparing the increase in macular pigment optical density from egg yolk, lutein versus equivalent doses from vegetable sources, have found that egg yolk produces a greater increase per unit of lutein consumed, making it an important complementary source even at lower absolute quantities. Corn, orange bell peppers, and green peas are meaningful secondary sources. Yellow corn in particular is rich in zeaxanthin specifically, the carotenoid that concentrates preferentially at the very center of the fovea, the point of maximum visual acuity, and that some evidence suggests is particularly important for preserving the fine central vision used for reading and detail discrimination. Now, the specific question of timing. Why before sleep? Why does when you eat this food matter? The circadian biology of carotenoid distribution in the retina is an emerging area of research, and I want to be honest about where the evidence is robust and where it is still developing. What is well established is the nocturnal function of the glymphatic retinal clearance system and the RPE's renewal activity during the sleep period. A serving of cooked spinach with olive oil consumed at dinner, 6 to 7 in the evening, would produce peak circulating carotenoid levels in the window of approximately 9 in the evening to 1 or 2 in the morning. The transit of lutein and zeaxanthin from the gut into the circulation following a meal takes approximately 3 to 6 hours, depending on the fat content of the meal and individual digestive variation. A serving of cooked spinach with olive oil consumed at dinner, 6 to 7 in the evening, would produce peak circulating carotenoid levels in the window of approximately 9 in the evening to 1 or 2 in the morning. That window overlaps with the early and middle sleep period, the hours during which the depth of non-REM sleep is greatest, during which melatonin is at its peak concentration, and during which the retinal glymphatic clearance and RPE renewal processes are operating at their highest activity. This is not a claim that the timing produces a dramatically different outcome than morning consumption. The evidence for timing-specific effects in the retinal context is not yet strong enough to support a claim of that magnitude. What it suggests, consistent with the emerging chronobiology of retinal carotenoid metabolism, is that the combination of evening consumption with adequate fat and cooking is a reasonable optimization of delivery to the retinal tissue during the period when that tissue most needs the protection and support these nutrients provide. For people who genuinely cannot eat a significant meal in the evening for any reason, acid reflux, preference, schedule, the most important variable is not timing but consistency and bioavailability. Daily consumption, cooked with fat at whatever meal is practical, is far more important than any optimization of timing. The circulating pool of lutein and zeaxanthin in the blood, maintained by consistent daily intake, is what feeds the retinal deposition process over weeks and months. The goal is building and maintaining that pool. I want to address what you should realistically expect from this dietary change and over what timeline, because the retina operates on a slow biological clock. Macular pigment optical density, the measurable accumulation of lutein and zeaxanthin in the macula, increases gradually with consistent supplementation or dietary enrichment. Studies measuring the rate of macular pigment increase consistently find that meaningful changes are detectable after approximately 4 to 6 months of daily intake, at doses equivalent to a daily serving of dark leafy greens. The deposition continues for up to 2 years before approaching a new stable level, with the rate of increase greatest in the first 6 months and slowing thereafter. Penthouse. This slow timeline reflects the biology of how carotenoids reach and accumulate in the retina. They are transported from the circulation into the RPE and then transferred to the photoreceptors through a multi-step process involving specific binding proteins and membrane transporters. The functional improvements documented in clinical trials, better contrast sensitivity, improved glare recovery, reduced photostress, typically become measurable around the 6-month mark in trials using objective measurement tools, and become noticeable to participants somewhat later. You are unlikely to notice a dramatic change in your vision in the first month of adding cooked spinach to your evening meals. That absence of immediate effect is not a sign that nothing is happening. It is the nature of a tissue that rebuilds itself slowly, and whose improvements manifest only after the structural changes are well underway. Penthouse. There are specific situations where the conversation about retinal nutrition requires individualization and physician involvement. Penthouse. If you have been diagnosed with age-related macular degeneration at any stage, the AREDS2 formulation that includes lutein and zeaxanthin alongside vitamin C and E and zinc is specifically indicated for individuals with intermediate AMD or advanced AMD in one eye based on the clinical trial evidence showing reduction in progression risk. Discuss your specific eligibility for this formulation with your ophthalmologist. Penthouse. The dietary changes I have described complement but do not replace the supplementation protocol indicated for diagnosed AMD. Penthouse. If you have been diagnosed with glaucoma and are being followed by an ophthalmologist, discuss the emerging evidence on lutein and retinal ganglion cell protection, not as an alternative to your prescribed intraocular pressure management, but as a potential complementary dietary consideration that your ophthalmologist may find worth discussing in the context of your overall management. Penthouse. If you are taking fat-soluble medication, including certain anticoagulants, the increase in fat intake associated with cooking greens in olive oil is generally modest enough not to represent a clinical concern, but transparent communication with your physician about any dietary change remains appropriate practice. Penthouse. For the majority of adults over 60 without these specific situations, a daily serving of cooked dark leafy greens, spinach or kale, with a tablespoon of olive oil or other healthy fat source, consumed consistently at dinner or at whatever meal is practical, represents one of the most specifically evidence-supported dietary changes available for protecting long-term visual health. Penthouse. It does not require a prescription, it does not require an expensive supplement, it requires a bag of spinach, a pan, some olive oil, and the consistency to make it a feature of dinner most evenings, rather than an occasional addition. Penthouse. The biology of your retina responds to what you feed it. Every night, while you sleep, the tissue responsible for your ability to read, to recognize faces, to see the detail of the world around you is either being maintained and cleaned in a nutritional environment that supports that work, or it is accumulating the oxidative and inflammatory damage that decades from now will show up as drusen on a retinal photograph, and eventually as the central vision loss of macular degeneration. Penthouse. The difference between those two trajectories begins with what you put on your plate this evening. Penthouse. If this gave you a genuinely new understanding of what is happening inside your eyes while you sleep, and a specific practical action you can take tonight that the research supports, hit the like button right now. It takes 1 second, and it tells this platform that detailed, honest eye health information for adults over 60 deserves to reach every person who needs it. Penthouse. Subscribe and turn on the notification bell. Every week there is one video here that goes beneath the surface of what most health content provides, into the mechanism, into the evidence, into the specific and actionable. Because that level of detail is what converts information into decisions that actually change outcomes. Penthouse. Leave me a comment answering this. How often are you currently eating cooked dark leafy greens, spinach, kale, or similar? And do you typically eat them with a fat source like olive oil? Those two answers will tell you immediately whether your retina is receiving the raw material it needs for its nightly repair work. Write them below. I read every comment and I respond when I can. Penthouse.